Hello everyone. This project is focusing on alternative splicing within parapatric populations of stickleback. Alternative splicing is a process by which a single gene can generate multiple mRNA transcripts, which may have different downstream functions, and this is thought to be a significant source of trait diversity. However, the mechanistic role of alternative splicing in adaptive processes is not well characterized. In order to study this, we utilize threespine stickleback, which are these fish that have repeatedly colonized freshwater systems globally, which you can see in this figure here, which highlights their distribution in the northern hemisphere, as well as the various model systems used to study this organism. The focus of this project is on this lake stream model. We utilize data collected from 4 distinct populations, 3 in Europe and 1 in Canada, all of which follow this general schematic of the Canadian population, where there's an inlet stream feeding into a lake with 2 distinct populations that overlap in the middle, but have trait variation and little to no gene flow between them. We hypothesize selection from habitat-specific parasite communities may be contributing to this divergence. In order to study the role alternative splicing has on this divergence, we measure differential splicing. The figure below is showing sequence reads mapped back to a gene. 8 of these reads map back to the second exon, while 2 reads skip it, meaning the second exon has a PSI, or percent spliced in, of 80%. We would then compare this PSI of the lake and river populations, and a change in the PSI greater than 5% would be a significant event. From this analysis, we have identified 162 differentially spliced genes, 32 of which were identified in multiple populations mentioned before. This is indicative of habitat-specific traits that are important to the environment in which the fish is found. In addition, 13 genes were identified with known immune functions. This highlights the parasite response as an agent of population divergence. These 2 gene lists would be excellent candidates for downstream molecular studies, teasing apart their functions in this adaptive process. Moreover, 4 of these genes were also identified as differentially expressed in a previous study, meaning their gene expression level was modulated. This highlights the molecular interplay of various mechanisms during evolution, and adds to the literature that these 2 mechanisms may function through different methods and pathways, which goes to the overall goal of this type of research, which is to better understand the process of evolution and how species diverge. Thank you for listening, and to my collaborators as well as my PI, Dr. Frédéric.